Hello viewers, welcome to this particular lecture on microbiology. In this particular lecture, I shall be discussing different aspects of biofertilizer, definition of biofertilizer, the different types of biofertilizer and their application and uses, and of course I will conclude with the different constraints of biofertilizer. So to start with, biofertilizer include a group of organisms which actually may be fixing nitrogen, solubilizing phosphate, producing hormone, and these organisms may be bacteria, cyanobacteria or sometimes fungi which grow in the rhizosphere, make the rhizosphere more productive. As because it is living organism, this biofertilizer may look like a granular structure because the soil is not homogeneous, and at the same time this biofertilizer, when it is sold in a packet, it will have some definite half-life or shelf life. Okay? So biofertilizer, as I was telling, may be of different types, and broadly they can be nitrogen fixing. The nitrogen fixing organisms can be free living like Azotobacter, Clostridium, Beijeringia. It can be symbiotic like Rhizobium, like Frankia. It can be associative symbiotic, that is the symbiotic nature is not perfectly expressed, which can be Azospirillum. It can be phosphate solubilizing bacteria like Bacillus megaterium or fungi like Penicillium, or it can be phosphate mobilizing biofertilizer, which include the arbuscular mycorrhiza, the ecto and endomycorrhizal fungi like the Rhizoctonia, or it can be Pizziza. And at the same time, biofertilizer can also be having the role of silicate solubilizing like the Bacillus or the plant growth promoting Rhizobacteria, PGPR. So these are the different types of biofertilizer. I will take one example from each. Rhizobium is a very important nitrogen fixing bacteria, which is of course having a typical symbiotic association with legume plant. And on the basis of specific association, there may be different strains of Rhizobium. But broadly, this Rhizobium is leguminosarum because it is growing in the root of legume plant and producing a typical nodular structure. This is the nodule produced by Rhizobium. And the Rhizobium as such is a rod-shaped gram-negative bacteria having the ability to fix nitrogen, supply it to plant, and thereby making the plant enriched in protein. The second one is Azotobacter. This Azotobacter has got a unique property. What is that? They have an outer slimy polysaccharide or extracellular polysaccharide with which they can adhere. And you can see here this particular white slimy outgrowth actually represents the EPA. And this particular Azotobacter also can be applied directly to the soil because it is free living nitrogen fixing organism. We come to the third one, which is Azospirillum. Azospirillum, as you can see, is a rod-shaped bacteria having flagella all around. So it is actually a peritrichous flagella. And this particular Azospirillum is also gram-negative bacteria, and it has the ability to grow in the soil which is close to a grass plant or gramine. So ideally, Azospirillum is helping in the growth of grass-like crop plant like paddy, like wheat, etc. So they are ideally providing nitrogen to paddy plant. And there is another important point. They also have the ability to produce a particular polyhydroxybutyrate, which is as an excretory product. Right. Now this particular Azospirillum, there are different species like Azospirillum brasiliensis, Azospirillum lipoferum, Azospirillum amazonensis, Azospirillum haloparferens, Azospirillum iracans. So these are different species of Azospirillum, and they also grow in different grass family members, enriching the rhizosphere with nitrogen. Okay. And Azospirillum can also be grown in petri plate, and they grow as discrete colonies like you can see here. This is the Azospirillum species growing in normal nutrient agar medium. Of course, this particular nutrient agar is special type because it is deficient of nitrogen, because if nitrogen is present, nitrogen fixation won't occur. So this is a very important case that you have to grow this bacteria in a nitrogen deficient condition, otherwise they will not grow. Now coming to the next group, which is cyanobacteria. Cyanobacteria can be unicellular, can be filamentous, but broadly as nitrogen fixer they are important as say Oscillatoria, Tolipothrix, Geotrichia, or they can be Anamina, Nostoc. These are different groups of nitrogen fixing cyanobacteria. Most of them, they grow as a free living nitrogen fixer, but some of them like Anamina, they grow in association with a particular water fern which is Azolla, and this Azolla is actually growing on a paddy field and supplying nitrogen to the paddy plant. At the same time, they may also grow in a particular rhizosphere of cycas plant, and that also produce nitrogen to this particular cycas plant. So that way I can say that this particular cyanobacteria can be free living, at the same time they can be symbiotic, growing in the rhizosphere or associated with the root of higher plants, or sometimes they may be endophyte as seen in case of Azolla and Anabina. Now this particular group is actually called phosphate solubilizer. Phosphate solubilizer meaning phosphate is added to the soil as superphosphate, plant cannot solubilize it. So these bacteria present in the rhizosphere will make that phosphate solubilized and supply it to plant, and this is what is called PSP or phosphate solubilizing Pseudomonas. This phosphate solubilizing Pseudomonas usually require a very low pH and they normally form an aggregate. You can see they are growing in an aggregate, so they are working together for the growth of the crop plant. And this particular phosphate solubilizing Pseudomonas can also be grown in a nutrient medium which is deficient in phosphorus. But at the end of the day, this phosphorus is made available to the plant and the plant can grow and the plant can actually produce more energy, so they can ultimately have a higher metabolic rate when this PSP is growing in their rhizosphere. Got it? Now coming to the different level of colonization in the VAM fungi. The VAM fungi actually is having two specific layers, the outer layer and the inner layer. The outer layer normally is serving as ectomycorrhiza and then they go in. And when they grow in actually, they sometimes can come into the inner layer of the cortex. In what way they are advantageous? Actually, this particular VAM will produce some mycelia and will acquire nutrients from the soil. And this particular VAM fungi can produce numerous amount of spore, and these spores, because they are produced outside in the air, sometimes they can be allergen and they can be pathogenic.
Sir, how does Gloma species serve as an endosymbiont when it is itself pathogenic?
I told you it is pathogenic, but to whom? To other animals or human. It is acting as an allergen, it is entering into the respiratory tract. But these spores normally, if it is below the soil level, it is not normally exposed, number one. Number two, this colony of mycelia, as I was showing you, this mycelia will actually take in more nourishment, make the root more healthy, make the plant grow in a better way. So that's why these VAM fungi or vesicular arbuscular mycorrhiza, they have different growth form which is beneficial to the plant, may not always be beneficial to human, but we are rest assured that we are not always exposed to it because they are underground. Okay. And as I was telling that the endosymbiont will grow inside, then they will produce arbuscle, and these arbuscles can sometimes be exposed on the surface. Otherwise, they can produce a lining or a layer outside the root, and this is in a way it is a protective layer, and at the same time it is drawing nourishment for the root plant. Clear? So this particular VAM fungi, this is the spore or this is the vesicle, which can be stained with a typical dye called trypan blue. So this is the blue vesicle of the VAM fungi, and there are different growth forms which can be mycelial, which can be absorptive. I was telling they absorb, and which can be rhizomorph. They go inside the plant, produce a thick mycelial mass called rhizomorph. They can produce thin tissues or mycelium, or they can produce spore as I was telling you, or they can produce thick walled sclerotia or resting spore. So that way they are polymorphic, different forms of worm fungi are there, and they are actually growing on the basis of a particular weather condition. So apart from glomus, there may be other species like gigaspora, and you can see these are gigaspora spore, and sometimes they are pigmented and they divide by binary fission, and that's why the replication rate is very fast. In this particular case, the endogone is producing a thick-walled cyst. Why? Because the external environment may be not friendly. So they are protected within that cyst, and whenever the friendly condition comes, the spores are liberated. So in this way, the reproduction mechanism of this particular worm fungi also can vary from one species to other. But ultimately, they are helping the plant to grow in a better way. Okay. We come to another group of bacteria which may be normal Bacillus, which may be certain fungi like mold fungi, which can actually solubilize silicate. And silicate, as you know, is a very important element present in the soil. And this silicate has catenation property, so it can produce innumerable compounds with other elements like oxygen, like hydrogen, etc. But this silicate, if it is present too much in the soil, then the plant may not grow. So in order to favor the growth of the plant, there may be some microbes which will convert silicate to silicic acid, or sometimes silicon dioxide, or sometimes zeolite, or sometimes other metallic silicates. In one way, this transformation of silica within the rhizosphere will make the plant grow in a better way, will increase the productivity of the plant. Okay. Coming to the next one, which is commonly a group of bacteria. This group of bacteria is called plant growth promoting rhizobacteria. Why? Because they are promoting plant growth being in the rhizosphere. And how? They include the different bacteria like Pseudomonas and Bacillus, which may not be pathogenic. They are present near the root, and they can produce different hormones like cytokinin and gibberellin. They can help in phosphate solubilization. They can produce indole acetic acid, helping in root growth. Sometimes they are producing specific iron absorption by siderophore. Sometimes they are producing antibiotics or molecules which will prevent the growth of other pathogenic organisms. Or sometimes they may have directly nitrogen fixing activity.
Sir?
Yeah.
In what way PGPR modifies the rhizosphere?
You can very well see the PGPR is a dynamic growth. PGPR modifies the rhizosphere in the following way. You can see here, it will modify by nitrogen fixation, that is enrich the soil with nitrogen. It will produce more hormone, which is growth hormone, helping the root growth. It will help in nodulation, which is a external growth. It will help in nutrient uptake, because I told you, say siderophore, ferric is converted to ferrous form and the ferrous is absorbed by the plant. So the soil becomes deficient in iron, pathogenic organisms cannot grow. At the same time, it can also produce an antibiotic which will prevent pathogenic bacteria like Xanthomonas. So that way it is also protecting the plant. It is an advantage. So this is how it can modify the rhizosphere. Clear to you? Now coming to the different benefits. Actually, these benefits are for the different biofertilizer. Innumerable number of benefits. I have only enlisted some of them. They have a moderately longer shelf life. Normally there is no contamination because they are highly specific, and usually they may be, not always, thermostable, and they can fight with other pathogenic organisms, and even they actually can maintain high cellular concentration within the cell without causing any pathogenicity. They may also be easily identified, and they can be screened, and they can be sold to people, and of course they can be applied in a very simple way. That is the reason why biofertilizers are important. But biofertilizer application usually is carried out in liquid form, and these liquid biofertilizers are really very important.
Sir, what is liquid biofertilizer and how can we apply it?
Yeah, liquid biofertilizer is basically, I told you, some specific microorganisms grown in a nutrient enriched liquid medium, and they are directly applied to the soil, or they are a specific mechanism which you can see here. They can be used as seed treatment. By applying liquid biofertilizer on the seed, you are enabling the seed to be protected from other pathogenic organisms, and at the same time you are providing nitrogen to the seed, so the seed will grow faster. So it is an easy way of protecting the plant and at the same time ensuring its faster growth. It can also be applied directly into the root when the seedling is being planted. Say for example the rice plant, you have to germinate the seed in a nursery and you have to transplant that plant. So when the transplanted plant is grown, it is dipped in biofertilizer. It's an easy method, and that way you can be rest assured the nutrients will be available. Thirdly, it can also be directly applied to the soil because it is liquid. You can directly add it, and that also a specific volume can be added to a specific mass. So not much problem is involved in it. So these are the three ways, that is either seed treatment, direct soil application or root dip. Okay. Now I was telling you the certain applications, as because they are not having different inorganic components, they are very good components for organic farming. Secondly, they will not really change the soil composition. There are sometimes report in the newspaper that too much synthetic fertilizer has actually changed the property of the soil, but when you are using biofertilizer, this won't happen. Thirdly, they are easily applied to seed, easily applied to the root, easily maybe applied to the rhizosphere. So these are very important point to be noted, and at the same time they are tested easily in laboratory condition and can be stored for a longer period, can be transported easily, and above everything they are not that much costly. But these are the brighter side of the thing. Whenever we see a brighter side, there must be a darker side. So there are certain constraints. So what are these constraints? There may be technological constraints. What are these technological constraints? First of all, you need to have a microbiological laboratory to produce a biofertilizer because it is a living organism. So this is an important constraint. You need to have specific equipments with which you will culture these organisms. It may not be available to a poor farmer. Thirdly, the shelf life, though it is said maybe up to 12 months, but sometimes it can be lesser. So you buy it and you have to use it quickly. If you don't use it, the efficacy is gone. There may be infrastructural constraints, that is the lack of equipments, lack of power supply, the space availability, all these may not be there, or adequate condition of inoculum storage may not be there. These are infrastructural constraints. It can be financial constraints. Why financial? You see, at the end of the day, I need to see that what I am gaining. I have applied the biofertilizer, but if I am not producing enough crop, then I am financially at a loss. So that is a financial constraint. Secondly, biofertilizers are ideal for a particular season. In too much rainy season, it will be washed out. So there is no actual use of biofertilizer during heavy rains. And at the same time, ultimately the return is not very high, return is less, relatively less. So these are financial constraints. There may be quality constraints. I told you, say for example, I can project that okay, this much of biofertilizer will give this much yield. But ultimately it doesn't happen. Why? Because it is a biological organism. You cannot guarantee, you cannot vouch for a biological organism, because applied to the soil, you never know in what condition they will be in and whether there is any negative organism there to stop its growth. At the same time, the awareness is not there. There may not be adequate awareness. People are not aware that okay, biological farming or biofarming or biofertilizer is such an important constituent which can actually protect the soil. And thirdly, the network is not there. People are not getting the supply. Say for example, the government counters are there, but this government counter every time the farmer won't know, so they don't actually use it. And at the end of the day, when they find out okay, it's the chemical fertilizer which I am getting in the rural base and that is cheaper to buy, I will use that. So ultimately, the farmer may not be interested because he doesn't know about the outcome. And at the same time, you apply a biofertilizer, you will expect that there will be a massive gain in productivity. But ultimately, when it comes to terms of productivity, it is not very high. There is not a visible change in productivity. But of course, the qualitative change is there. You can very well understand, okay, by using biofertilizer, you will get a crop which is not toxic, which will not have the toxic implications of pesticide, which will not have the toxic implications of synthetic organic fertilizer. Or at the same time, you know you have protected the soil because the soil is not getting contaminated. But who thinks about it? Because everyone is running after money. So the farmer, when he sees that, okay, I am not gaining by applying biofertilizer, so naturally he will not be interested in buying it. So these days we see that biofertilizer movement has to be taken forward by young people. They should explain it to the far corners of this country, telling that these are the good points of biofertilizer and you should adopt biofertilizer not only for people but for your own existence, because you never know that synthetic pesticide or fertilizer in the long run can cause tremendous damage in human beings, in cattle, which has to be avoided. And the time has come that all of us are aware of this and we shift to biofarming, which is ideally by using biofertilizer. Thank you.